Okay, so hello, good afternoon, everyone. My name is Ankita. I am a TA for this course on Basics of Biology by NPTEL. We are now starting off the live interaction session for week 6. And on today's agenda, we are first going to go through a recap for this week. Then we'll discuss the assignment, the questions that were already given for this assignment, and also some problems from week 7. And overall, we'll cover some different topics for this week. So to first go through a recap of the topics that were covered, we are going to use the lecture notes that were provided as part of on the portal as part of this week's reference. So in the first lecture, we had actually seen, and if you've gone through it through the videos and through the lecture, you will see that basically what the topics were focused on was the central dogma of life. So the initial topics that we have covered till now, they involved evolution, cells and their understanding, biomolecules, and currently we are discussing cellular processes. So this week actually covers cellular processes, and it is the the first part of cellular processes. So under that, we are looking at all of the different aspects of life, and that of course involves self-growth, self-renewal, that is the ability to survive and the ability to grow and the ability to survive and to recover from any damage that might occur. Then the endogenous ability to produce energy, which means to metabolize. Then the ability to move, exception of course being plants. And finally, the ability to replicate, to produce progeny, that any living organism species. And okay, so we are going through a revision or a recap for week 6, and as part of that, in the first lecture, we have actually discussed the central dogma of life, and as has been discussed extensively, the central dogma basically consists of 3 major steps or 3 major types of biomolecules, which are obviously DNA, RNA, and the proteins. So DNA, the master molecule, is responsible for containing all of the genetic information that would allow an organism to transfer or to carry it forward towards its progeny. RNA is the second step or the second stepping stone, you might say, after DNA, which is executing or it's actually decoding what is the information that is present in the DNA so that it can be further translated or can be further made functional by use of proteins. So as part of that, as part of the central dogma, the processes that we refer to in this case are obviously replication, which is where DNA is being used as the template, and from that we are getting the duplicates of DNA, or we are getting more and more DNA molecules which would be required so as to sustain more and more cells. The second process, which is where you use the DNA as a template and then synthesize the second molecule, second type of major biomolecule, which is RNA. That process is occurring via transcription. And finally, in step 3, which is where the information present within the RNA is decoded or it's converted or it is translated into proteins, that is known as translation. And these are the 3 major steps which are together constituting the central dogma of life. Then these processes were discussed in quite detail. Where exactly do they occur? Like, for example, replication occurs during the S phase or the replicative phase or synthesis phase of the cell cycle. Then transcription, it is occurring like the cellular localization for transcription is within the nucleus, whereas translation, the process of translation occurs within the cytosol, because obviously ribosomes are present in the cytosol, and some of the ribosomes are free moving, whereas some of them adhere to the endoplasmic reticulum, which is where we get rough endoplasmic reticulum or RE. Yeah. So these were the 3 basic steps that were initially discussed. However, there are some exceptions to the central dogma as well, where the first major example is of reverse transcription, whereby RNA is used as the template and from that DNA is synthesized. So that is a special circumstance. It's not always occurring within any living being. And most common example of the same is in case of viruses, that occurs very commonly in viruses. And the enzyme that works under those circumstances is an RNA dependent DNA polymerase. And there are also some other special circumstances which you can see here, like where RNA is synthesized from RNA, or directly DNA can be converted into protein. So there are exceptions to this. So in case of RNA replication also, like I said, for viral genome, you can often see that. And in an in vitro cell-free system, you can directly take a DNA and can synthesize protein from it. Usually within a living organism, this would not be a feasible option. Then the final fourth uncommon or unusual possibility is from protein to protein. So these are also organisms that we had initially discussed during the week one discussion sessions, where we were talking about classifications and the diversity in living organisms. There we had seen that there are some exceptions in the living world where you cannot very distinctly, you cannot classify organisms, and some exceptions to this included, of course, viruses, but also prions. Prions are proteinaceous infectious agents, and they are an example of an exception to the central dogma of life as well, whereby protein molecules are able to form their not really progeny but their copies, and those can further carry out the they can further infect other organisms and can sustain by themselves. So they are also an exception to this central dogma as well. So after that, in this lecture, there was quite in-depth discussion about the process of DNA replication. And I won't be going into details, obviously, because there was a lot of material that was covered in this week. But overall, we'll just recap what was discussed. Between them. And this process is obviously known as hybridization, and it brings forth the major one of the major properties of nucleic acids, whereby there is a complementarity between the two anti-parallel strands. Now, this complementarity has been demonstrated here as well. Then comes what are the factors, or what are the contributing partners that actually execute this entire process of replication. So there are a huge number of proteins that function in each of these biological processes. In case of DNA replication, the machinery that is involved, it starts with primase, which is the enzyme that Pol alpha primase, it's commonly known as Pol alpha primase, that brings about that actually synthesizes a short piece of RNA at the site of replication, and it initiates the process. Then comes polymerase, and obviously, as the name indicates, it is involved in polymerizing the synthesis process. It keeps on adding nucleotides to the pre-existing segment, and it is responsible for the elongation step. And its different activities, polymerase, exonuclease, and proofreading activities are also discussed here. Then comes DNA gyrase, which is required for unwinding. When you have double-stranded DNA, you do not completely separate the two strands, obviously, because there is a lot of energy that is required to do the same. So as the replication fork moves, the DNA that is coming like the DNA that next would undergo, the portion of the DNA that would next undergo replication, that has to be unwound. So gyrase, it moves in concordance with the entire replication machinery, and it keeps on unwinding DNA in front of it so that the replication machinery can move. Then comes ligase. Ligase is required because after you have synthesized not you, the replication machinery has synthesized a portion of the DNA. The fragments that are synthesized, they need to be joined, particularly like we have previously discussed, the Okazaki fragments, they need to be ligated to each other. Then comes SSB, or a single-stranded DNA binding protein. Like we said, the gyrase is constantly unwinding DNA, right? So when it does so, a crucial step to ensure that DNA remains unbound is to recruit these proteins, and it reduces the possibility that again gyrase needs to come in and it needs to spend energy, unwind DNA, and that would of course be a interrupting step in the entire process. Then each of these steps of replication were discussed. fork that is formed. You have helicases and topoisomerases that are present, and they are unwinding DNA and resolving the supercoiling that is present within the DNA prior to the movement of the replication fork. Then comes the process of elongation, which is obviously executed by the key enzyme involved in this process, which is DNA polymerase, and it keeps on adding DNTPs and extending the length of the polynucleotide chain by inserting nucleotides that are complementary to the nucleotide present in the template. That is where the complementarity it comes in, and that is where Chargaff's rules and all of these processes we have discussed before, all the properties we have discussed before, they become extremely relevant. And this is the elongation or extension step that is further carried out by the DNA polymerase as well. And obviously extension is different for the leading and lagging strands because of their orientation from 5' to 3' and 3' to 5'. For the lagging strand, you will get Okazaki fragments which need to be ligated by DNA ligase. And then comes the termination step. So termination is majorly done by the ter sequences, which is where the replication process would suddenly or like abruptly it halts, and the extension is no longer as it was done previously. And finally, you go through proofreading and that the sanctity of DNA is maintained. That from one parental organism to its offspring, there should not be an immense change because it would affect the survival of an offspring, right? And if the parental organism has acquired some beneficial properties because of some sort of change in its genome, then those beneficial adaptive properties should be carried forward, and therefore DNA replication needs to be done very carefully. So there are mechanisms like, of course, the enzyme itself has its own proofreading activity, but there are also mismatch repair. There's an entire system that operates inside the cell, which is known as the MMR, mismatch repair system, which operates in absolute conjunction with this replication process. And throughout that S phase of the cell cycle, it keeps on checking that, each base that has been inserted, for example, if there is A in the template strand, there should be a T that is inserted in the next one, in the complementary one, or in the daughter strand. So these mechanisms, they are very relevant and they are very much operational. But despite these checkpoints, also we do see that there are a lot of mutations that are acquired, and that is something we cannot really bypass. So DNA replication was then summarized here, and that completes the first and second lecture for this week. In the second lecture, what was majorly discussed was PCR. So continuing with the theme of nucleic acids, PCR or polymerase chain reaction as is the technique which was developed by Kary Mullis, is very simply put, it is a method of amplifying, or it is a method of increasing the amount of template DNA. So if you the question would be that why do you at all need it? You would need it because the initial amount of DNA that you might get, for example, during an experiment, the amount of DNA that you might get is very small. It's very little to really study or analyze anything or to do anything with that at all. So you need to, more often than not, you need to increase that amount. And for that, this extremely brilliant technique of PCR is used, and it is used thoroughly in like every single lab, almost every lab you can think of. So that is what you're doing. That's what's been explained here as well. You have double-stranded DNA molecules with like approximately same size and sequence. They are increased or they are amplified by cycling, basically thermocycling, which is where you continuously or cyclically you keep on altering the temperature until you get an increased amount of DNA. So these were the proteins that were involved in replication. So basically, if you think about it, there are three phases, right? For synthesizing DNA, you have a step where you are initiating the process, you are increasing the length, or you are carrying out an extension or elongation step, and then you are stopping it at a definite point of time. So the process of polymerase chain reaction, these are the different events that occur and it was a revolutionary discovery in the in one of the most relevant and revolutionary discoveries in the recent past. So coming to PCR, like we have already said, it is a repeated cyclic reaction where you have one single template and that gets amplified, that needs to be separated. To each of these separated single strands, a small primer goes and binds, and that binding step is known as annealing. And the bound primer is then elongated by the process of, or by virtue of complementarity, base complementarity, it is elongated. And that this is repeated a number of times, around 40 times, 30 to 40 times it is repeated to generate a huge amount of DNA, which you can then use for whatever reaction you want to set up. So the amount of DNA which is amplified and which is synthesized is often calculated by this formula, where you have C, the final amount of DNA, C0, the initial amount that you had, multiplied by 1 plus E, the efficiency to the power raised to the power n, where n is the number of cycles. And that gives you an approximate idea of how much DNA you would initially get. So this is an example of the type of PCR reaction which is usually set up in research laboratories. So you have three stages, as we have discussed. In step one, you carry out the initiation step, which is where you set up a reaction at around 95 degrees for approximately 5 minutes. What does it do? It ensures that DNA is completely denatured. After that, you enter the cyclic phase. Now, this cycle will have denaturation, annealing, elongation repeated multiple times, a number of times. So the double-stranded DNA is denatured, and this is for around 15 to 30 seconds at that very high temperature. Then there is an annealing step. Now, for annealing to occur, you need to reduce the temperature so that these two molecules, so that the template and the primer, those two molecules can come close to each other and they can bind to each other, and the energy required is feasible for doing so. So that temperature is usually somewhere kept around 50 to 60, 65 degrees, and it allows this will depend upon the type of primer which is being used, and like that is a whole other topic of discussion, how a primer is designed, what should be the parameters that are kept in mind before you synthesize or before you design and synthesize primer for a particular template DNA. A lot goes into that, and that also affects the quality of your PCR. Then the primers can bind, and that annealing time is like approximately 15 to 30 seconds. And finally, after these cycles are completed, then there is elongation, and then after that you allow it to stay at that temperature for around 10 minutes. So during this elongation step, you can see that polymerase is adding new DNTPs, and the average efficiency of polymerases are assumed to be somewhere around 1000 base pairs per minute. So you carry out 35 to 40 such cycles, and after that, after this cycle is complete, you give another 10 extra minutes for any incompletely synthesized or partially synthesized DNA to get duplicated, and then you finally hold it at 4 degrees for like an infinite period of time, and that completes the step. Then we come to the basic idea of PCR. It's given here. Now coming to the requirements for this process, the region that you're trying to amplify, you should have sequence for that. The primers, their approximate size, a thermostable DNA polymerase, which is, you can say, the most important factor for a PCR reaction. It has to be an efficient, high fidelity polymerase, and here you're talking about Taq polymerase, which does not get inactivated at that extremely high temperature. And that brings us back again to something we discussed in the first session about how extremophilic bacteria are extremely relevant in this context, because Thermus aquaticus is the is the organism from which this enzyme was isolated, which makes it obviously very relevant. Then a thermal cycler, the machine which can do this. So this is an example of a thermal cycler by applied biosystems, where you can set up a cycle of such temperatures and it will carry out as number of cycles as you have requested in that period of time. So this is an instrumentation. We don't need to get into it. Then the amounts required were discussed, and the template DNA. So template DNA can either be the genomic DNA. It can be some fragment you might have isolated. It can be a circular plasmid. It can be viral DNA. You can have some patient tissue sample from which you have homogenized. You have gotten single cells or whatever. You have ruptured the cell. You have taken the nuclear fraction. Then you've isolated DNA from it, and you're using that as the template. So the starting point can be different, but then PCR, as you can see from here, it's like hugely vast. Its applications are extremely vast. Then coming to the primer. So you have two sets of primers. You have forward and reverse primers, which are complementary to the template sequence, and they allow the initiation process of the synthesis process to begin. Then, like we were discussing about Thermus aquaticus, it is this microbe is found in hot springs in the Yellowstone National Forest, and the temperature is approximately 176 degrees Fahrenheit. And tacpol is capable of being functional at these extremely high temperatures, because for this microbe it is the ambient temperature. So the microbe has evolved in such a way to adapt to extreme temperatures, and we are just using its enzyme for our own use. And the optimal temperature is somewhere around 72 degrees centigrade. Um, But tacpol is not a one-stop solution for all problems. So its major issue is, as has been mentioned here, it lacks this exonucleolytic activity, which is why it has a tendency to incorporate a lot more errors than you can afford in such a sensitive reaction. Many a times it misincorporates as many incorrect bases as 1 in 1000, which is a very high rate. If you have like a 15 kb DNA, which is of course a huge fragment, but just assuming, just a statistical example, then it will incorporate 15 errors, which is immense. So here an example, you can see a 400 base pair target will have like almost 33% of the molecules will contain an error in 20 cycles, and this error distribution will be random. So you cannot, even though a tacpol has, it is often, I wouldn't say often, but it is used sometimes. But there are better high fidelity polymerases that are preferred in the like recently. So one example is the fusion, like it's written as PFUSION, fusion enzyme from Pyrococcus furiosus. This is used. It has this exonucleolytic activity. Its error rate is very low, which means that its fidelity is very high, and it avoids primer dimering. Then the reaction setup is as follows. You can see usually a 50 microliter reaction is set up with a forward and reverse primers, MgCl2 or magnesium chloride, because it's a requisite coenzyme for cofactor for this enzyme. DNTPs, the polymerase, buffer, water. So that's what you see here. This is how the reaction is set up. You have to optimize it. These are some optimization techniques that are given, and after that you analyze the reaction that you have gotten. So here you can see these are amplified fragments which are shown. So you usually the quickest way to actually study it is that you use an intercalating dye such as ETBr, which is ethidium bromide, and then use UV light, because that sort of it leads to, it lights up the DNA, and you'll be able to just visualize it directly on a transilluminator. Like the image that you're seeing, that's what has been done. On the base, there is a UV light, and ETBR intercalated DNA, it lights up and you can see where the fragment is present. And here you have the molecular weight marker. And very low percentage gel can also be used depending on what is your need. So why PCR at all? The speed, the ease of use, how robust it is, and of course its immense sensitivity. But it is again not the solution to all our problems. You need to know the sequence information. What if you don't? You can't. You need to know the sequence to design a primer and at all set up a reaction. So that's a major drawback. There is often infidelity with enzymes like tacpol. The length or yeah, the length or the number of bases of DNA that can be amplified, that is also somewhat limited, and genome sequencing projects are not cannot be clubbed with this. But its applications also are quite immense, like for molecular identification, DNA fingerprinting, which we have quite we've heard about it, like, right, in like during crime, like if you find some sort of a hair, or you find a saliva or some tissue from that individual, then that is often used to figure out who was there at the crime scene. Then for a determination of paternity of for a child, these are some very commonly heard of applications. And apart from that, obviously, you can see these are more extensive applications of it. In sequencing, it is huge in genetic engineering for site-direct mutagenesis, to check to check whether for example, if you're trying to overexpress a gene, if you want to see whether it has undergone overexpression or not, one would be you would look at the expression, but apart from that, how many copies of that gene are present, that also you can check using PCR. In food science also, it is quite vastly used, and in food microbiology, it's immense applications have been discussed in details as well, like the microbes that are there, how they are checked using qPCR, using multiplex qPCR, etc. You can go through it. And in medical sciences, how it was used, example including the amount of HIV in blood could be measured. Then it has been used for the multiplex PCR technique was also used on a single sperm cell to analyze what are the products of recombination that occurs, like meiosis that occurs in a living individual. In infectious disease also, it's used, like we discussed, HIV-1, Hep B, C, human papilloma virus. All of these are, even if you look at a real life example, all of the tests for the COVID-19 virus infection, they were all RT-PCR based. So RT-PCR obviously is also a PCR technique. Yes, the strategy is different. It uses RNA as the template because the viral genome was different under those circumstances. But then, yes, the long-term intent or the execution remains the same. So apart from that, we also see its role in blood screening. For serological screening, you can check. In plant science also, it is quite extensively used. What is the species of the plant? Because there are a lot of known relatives of some plant species that are often present. So you need to, many a times that is used to relate to identify which family a particular plant belongs. Then for plant microbe interactions, in plant tissue culture, in veterinary parasitology, like some pig diseases, some bovine diseases, etc. In forensic sciences, like we said, for criminal investigation, in DNA fingerprinting, if there are multiple suspects, you would need to have a technique in place which can narrow it down to one or two people. And one of those ways is to actually look at the VNTRs, or the variable number of tandem repeats, using some sort of a specimen that could be left behind by that individual. And you can check that out. Then for parental or paternity testing, this is also used. Short tandem repeats are looked at, and of course, its major application is in research. You cannot imagine 90% of the research labs around the world to function without having a PCR setup in one way or the other. So it is a, like I said, a revolutionary discovery by Caramelis. And here are some examples of its application that are provided as well. So that basically covers the third lecture for this week. Then coming to the fourth lecture, we looked at transcription. Fourth and fifth lectures looked at transcription, while the next two looked at translation. So in transcription, as we have already discussed, it basically is the process. When you heard about transcribing, like you hear of people who write down what, like for example, in court, there is one person who writes down whatever is being said. So that person is often said to transcribe. He's putting down what is the information that is present and decoding that information, right? So that is basically the process of transcribing. Here also, in context of a living cell, a similar process is done, where the information present within the DNA is broken down in the form of RNA. So the types of RNA, however, can be quite extensive. Like you can see here, we have tRNA, rRNA, and mRNA. And protein synthesis is from the messenger RNA only. It's called the messenger RNA for this purpose because it is carrying the message from the DNA to proteins. So yeah, so that is the process. And yeah, here what we talk about is a transcription unit. That is, okay, so not the entire genome can undergo transcription. There are regions of the genome often known as coding regions. Within the coding regions as well, there are some portions which are actually the regions which get transcribed, and they form an mRNA or RNA tRNA. The remaining portions are majorly involved in the regulation part, in controlling the process of transcription and controlling the expression of those regions. So the part that actually gets transcribed is the transcription unit. For eukaryotes, we have monocistronic. In prokaryotes, it is polycistronic, where you will have one coding sequence giving more than one polypeptide. In case of eukaryotes, it's the exact opposite. So you have a starting site, a transcription start site or TSS, from where RNA polymerase will bind and start. Then there is that is actually known as the +1 site. Upstream of it, you have -1, -10, -35, which are significant regions, and downstream of it are the plus sites, which are beyond the transcription start site, which is where the information, which is from where the mRNA will actually be synthesized. So yeah, so that's what we look at. And then the two types of strands, like since obviously we know that DNA is a double-stranded DNA, so just use this figure as a reference. So this is DNA. From here, you have one coding strand, one non-coding strand. The easiest way to remember this is that the code of the coding strand is actually taken forward. So if you have ATGC in the DNA, then that will be exactly the sequence of the RNA. In RNA, it will be AUGC. The non-coding strand is the complementary one, which is used as the template to insert bases against it. Similarly, the coding strand is also called as the sense strand because that is the sequence that makes sense. The non-coding is the antisense strand. It's easy to get confused between the two, but just use this logic to remember that the coding strand carries the code, or it makes sense. Let's just think of it that way. Now coming to the process of transcription, as you can see from this table, there are a lot of differences in the process among prokaryotes and eukaryotes, mainly the type of transcriptional unit, its location. So obviously prokaryotes do not have compartmentalization per se, even like the distinction, what should I say, the intracellular distinction or compartmentalization is not at all present there. So in case of prokaryotes, it occurs in the cytoplasm, but in eukaryotes, it's in nuclear process occurs. Process is coupled in case of prokaryotes, whereas there are multiple mechanisms that actually operate and that fine tune whether transcribed a portion of the DNA or a transcribed newly synthesized RNA, does it immediately undergo translation or do you wait on it? So there are a lot of checkpoints that operate, and the requirement for transcription factors, the formation or the structure of the RNA polymerases, and the number of RNA polymerases, these are all like some distinguishing factors in the two types of transcription. So here first we are talking about the prokaryotic process. So there you see that there's a single RNA polymerase with alpha alpha beta beta and sigma structure. Each of them have their own distinct functions. This is the promoter site. Now, like I said, the +1 site is where transcription would start. Now above it, -10 and -35 sequences are two consensus sequences, 6 base pair consensus sequences. When we say consensus, we mean that these regions are usually seen to be majorly the same across many different organisms, which actually brings forth their potential significance. That if you are irrespective of what organism it is, if you are seeing that these regions have 90%, 90 to 95% of the time they have the exact same sequence, then some function could potentially be attributed to it. So these are consensus regions. Then the different different steps or different stages in transcription would be the binding of a template, the process of initiation, elongation, and finally termination. So first step is the binding of RNA pol to template DNA and the process of initiating transcription. Then binding of RNA polymerases and chain initiation are considered. Then there is formation of an open promoter complex. So after the polymerase has bound, there is a local melting which occurs, which strengthens the what should I say? The strengthens the binding between the enzyme and the nucleic acid, or the um, the DNA, and that is called as the open promoter complex formation. And there's a local melting of approximately 10 base pairs. And after this binding, it's basically that this is the commitment step, that now the cell will certainly go, like it is now committed to the process of transcription in case of prokaryotes, of course, in this case. So that is what we see. Then chain elongation is occurring, and that is where you keep on adding more and more nucleotides, and the chain keeps on elongating in the 5' to 3' direction. So that is carried out in a transcription bubble. And then comes the chain termination step. So towards the end, when it has reached the end of synthesis, then it releases a complete product, and the RNA chain is not the RNA chain, the RNA synthesized, it is separated from this DNA, so no longer it remains bound. And this process of termination occurs via two different or two possible approaches. One is that of intrinsic termination, which is without any further resistance, and we'll actually go into details about it. It actually depends upon the sequence of DNA in that region. And the second one is with help of an external factor. In this case, it is the Rho factor or hexameric protein, which kind of it rolls, and it basically pulls the synthesized RNA away from the DNA, and it releases it. So this is the process of intrinsic termination. So if you look here, you will see that it does not require any additional proteins, right? So it has a GC-rich hairpin structure. Why GC-rich? Because the triple bond that is formed between G and C, that actually makes it much stronger. And what it does is it preferably these two would, like, instead of RNA DNA hybrid, forming a hybrid within this within the RNA product, it's more energetically feasible, and therefore it is preferentially chosen over binding to the DNA, and that allows its release from it as well. So sorry. So that's what we see here as well. Then the steps in intrinsic termination are explained here. Like we were saying, two inverted repeats are present, which result in the formation of such a hybrid or such a hairpin structure. And because of this, its bond with the DNA gets broken down, and RNA molecules are separated from the DNA. The second possible approach is using a row dependent strategy, whereby this hexameric protein, it comes in, and what it does is it first binds to the RUT site, which are cytosine rich residues, and a row, it basically chases the RNA pol. So for example, here you have the polymerase, row will chase behind it, and what it will do is at some point it will catch up, and it will pull the synthesized RNA away from this DNA hybrid and would release it effectively. Both these mechanisms can operate, and there is no like hard and fast rule as to which one would be present in which organism. Then coming to the eukaryotic process, one major difference, as we have previously also discussed, is that the number of additional factors that are present in case of eukaryotes is much larger. Just a second. Are much larger in case of eukaryotes. So there you have transcription factors that come in, and they play actually a much bigger role than the eukaryotes in this case. So here you have than the only the enzyme, instead of the enzyme in case of eukaryotes, the number of transcription factors have a more relevant role in fine tuning the process of transcription throughout in each and every step. But apart from that, the enzyme, the key enzyme, of course, RNA polymerase, which is a DNA-dependent RNA pol, that is also of three different types. And you can see here that they can be divided based on the number of or the type of function where they can synthesize RNA, mRNA, or tRNA. Their sensitivity to alpha-amanitin, which is used to check what are the different types. So you have resistant, sensitive, less sensitive, and their cellular localization or intracellular expression site. So within the nucleolus, nucleoplasm, they can be differentially expressed. So yeah, that's pretty much this. And mitochondrial and chloroplast RNA polymerase are said to be similar to bacteria as well. In case of eukaryotes, the promoter is different. You have an upstream promoter element. You have a core promoter element, and you have some promoter elements. It's more of a fragmented promoter in case of eukaryotes. Some portions are very close to the transcription start site, which is what is known as the core promoter element, where the RNA poly gets attached and forms the complex, and from there, synthesis can occur. But there are also a lot of sequences which are located quite far off, and those are the upstream promoter, or sometimes they can be downstream promoter elements as well, but mostly those would be classified under enhancers. So promoter elements you can majorly see as upstream ones. So transcription initiation, I'll not get into much details about this, but you can see there are so many general transcription factors which are present, starting off with TF2D. This is quite relevant to actually remember, which is why I'm mentioning it like in isolation. So TF2D, A, B, F, E, and H. This is the sequence in which they get recruited as well. So that is significant too in our case. And that is what you can see here as well, that the process of initiation involves their incorporation one by one into the entire transcriptional machinery. And then comes the process of termination. So RNA-pol2, it requires these genes, they can be huge, they can be extremely long, but towards the end, the termination process will take place, and that can occur again by the row dependent termination process for pol1. For pol3, it is independent, and in case of pol2, it is usually coupled with a processing event where the 3' end undergoes cleavage and polyadenylation, which we'll also discuss as part of the post-transcriptional processing events. So that is the termination process, and we'll now move to the next one. Yeah, we discussed this already, eukaryotic promoter. It's a little repetitive, just a second. Yeah, so post-transcriptional processing, here it's written as modification, you can call it processing, that I think is more accurate. But basically for mRNAs, there are some conserved, you can say some conserved processing steps that always take place in order to convert the synthesized mRNA into the more functional form. It's often called as the mature processed mRNA. So it needs to have a 5' cap. It needs to have a polyadenylated tail at the 3' end, and it needs to have introns removed from the sequence. So because we said that there are some regions which are coding, some are non-coding, so you need to remove those interrupting sequences, which are otherwise known as introns, from this mature mRNA. So the first step is the capping step, where you can see that the methyl guanine triphosphate cap is added. It occurs as soon as transcription has started, and the nascent mRNA is emerging, the cap is attached to it. And this I'm not getting into much detail how it occurs, but the capping step is extremely crucial, because that will indicate that the transcription has occurred with absolute fidelity, and this is an mRNA, it marks it for further processing. Second is the addition of a polyadenylation tail. So it is involved in, like I said, in export, it increases the mRNA stability, and it is a very crucial recognition signal for binding of other translational factors during the initiation of that process. And this process, it is carried out by one very specific enzyme, which is a template independent RNA pol called as PAP, or poly A polymerase. And the third and very, very crucial step, which is absent in case of bacteria, is the other two are also absent, but this process is known as splicing, where you splice out or you selectively remove any interrupting sequences, otherwise known as introns. So these are non-coding nucleotide sequences that are not protein coding, and therefore they need to be removed. And these are removed, and additionally the exonic regions are ligated to each other. So there are four classes of introns, group 1, group 2, spliceosomal introns, and introns that require ATP for splicing. So here we see group 1, group 2 introns, the different mechanisms that are operational for each of them. And for these two, you have two trans-esterification reactions, which is what you see here. Esterification reactions are occurring, and the it's removed or excluded or spliced out portion is removed in the form of a lariat, which is like a closed kind of closed structure, which is particularly seen for the group 2 introns. So that is what we see there. And then we have alternative splicing. So um, under normal circumstances, splicing is a very crucial step. So if you have one particular mRNA, you remove all of the introns from it, and what you do is you join the exons and you have the mature or processed mRNA that proceeds for translation. That is like the default setting, you can say. However, because eukaryotes have learned to be efficient, what is done is that often, many a times, from the same mRNA, what you do is you create alternative or uh, different possibilities or different alternative ways in which this processing can actually occur. And as a result of that, the splicing process can also occur differentially and or in different manners. One very, very relevant example of this is sex determination in Drosophila. And also another common example that you can take back is the processing which is done for immunoglobulin genes, which are genes from which you basically get antibodies. So you have the heavy chain and light chain. This will be a part of week 7 as well. So you have this particular sequence or this particular mRNA, and that not mRNA in fact. Okay, and premature mRNA, let's call it that. And from there you can process it in different ways. You can choose different combinations which are mutually exclusive of each other, which yield different types of gene products, and that is where you get a lot of variation. So that is another way of very smartly or very strategically using one template for a number of different functions. So that's a brilliant strategy. Then coming to yeah, here we see the immunoglobulin heavy chains. Then the production of different hormones, etc. These are like alternative examples for the same thing only. Then comes the processing of rRNA, which is the ribosomal RNA. We talked about mRNA processing before that. So here, in case of rRNA, you have two basic steps where there is cleavage and there is base modification, which is RNA editing. So RNA processing in bacteria can occur. So it's described here in elaborate detail. In case of eukaryotes also, you can see pre-rRNA, which gets, you have 18S, 5.8S and 28S rRNA as part of a longer RNA. From there, this needs to be chopped off and the mature RNA needs to be produced. And then we have the tRNA, which is the you can call it like the executor molecule, because it brings in the amino acid which needs to be incorporated in the newly synthesized polypeptide chain. When you are decoding an mRNA, when you are synthesizing a protein from it, this tRNA plays the most important role. So it brings in the charged amino acid and allows its incorporation to actually be executed. And it involves using some enzymes, or not using, the role of some enzymes like RNase P and RNase D, and the 3' so carried out. Finally, to the last topic for this week, after which we'll move to questions. So as part of the you could say the last step, so-called last step of central dogma, it is translation. So translation is the process of protein synthesis from the using the mRNA as the template. So you have the translation machinery, the genetic code, the mechanism, how amino acids are activated, the different sub steps such as initiation, elongation, and termination. And finally, after translation is completed, the post-translational modifications, which are the easiest requirement Yeah, so the messenger RNA is its first major requirement. And if you look at the structure, we talked about how there is a cap and a poly A tail, which is added to the mRNA, which is undergoing processing. But after that, we have, apart from that, we have a 5' UTR or an untranslated region and a 3' UTR, a ribosome binding site, which is not seen here, but that is where the ribosome would bind and it would allow this particular mRNA to undergo the process of translation. And then we have the coding sequence, and it has 3 reading frames as well, which will come after we discuss this genetic code. And finally, the entire region, this is called as the open reading frame. So if you see, like the from the point of from the translation start site till there is a stop codon, which is basically the region that undergoes translation, the process of translation. That entire portion is called as an open reading frame. Open reading frame, basically this is where the ribosome, the entire region or the entire stretch of ribonucleotides that a ribosome would read and would translate via incorporation of amino acids. That is what it basically means. So that is what we see here. And then we have the genetic code as well. So genetic code obviously is made up of the same 4 bases in this case, because the RNA is being read. The bases are AU, G, and C, not AT, G, and C. And from there you have all of these 64 possible combinations or 64 possible amino acids that exist. But since only 20 of them are known to be naturally present, from here, one of the major and one of the most significant and again very revolutionary understandings about the genetic code was that it is degenerate, which means that there are multiple codons which code for the same amino acid, like UUU and UUC. Both of them, they code for phenylalanine. For leucine, you have 4 different codons, right? For arginine, you have 4 different codons. So that is the degeneracy of the genetic code. So that's what is mentioned here as well. First of all, the genetic code is triplet. 3 bases correspond to 1 amino acid. There is a start codon, there is a stop codon. It is unambiguous. That means that the code is only for 1 amino acid. So you will never find UUU coding for both leucine and phenylalanine. It only codes for leucine, or in this case, it only codes for phenylalanine. There is no comma. There is no gap in the code. Each and every nucleotide is red, ribonucleotide is red, and against it an amino acid is inserted. It is degenerate, and we discuss this property like UUU and UUC. Only exceptions to this are tryptophan and methionine. Yeah, so sorry, I was getting disconnected for some reason. So it is degenerate, like we were saying, and only tryptophan and methionine are exceptions to the process of degeneracy or the possible degeneracy of the genetic code. They are only coded by a single codon. Then other properties are that it is non-overlapping. So each, you will never, the ribosome will never read it as AUG, UGC, GCU in this manner. It will take the first 3, it will shift an entire frame, it will read the next 3, and so on and so forth. So the genetic code is universal. It is common for all life forms, but there are exceptions to this. So for some microbes, you will find this, like for example, UGA is a stop codon. We know UAA, UGA. These are stop codons, but what can it do? It can code for tryptophan in case of, for example, here it's shown mycoplasma, for spiroplasma, and many, in a lot of cases in mitochondria also we see a lot of difference. Some of our stop codons are sense codons in their case, and vice versa. So you will find some exceptions to this, but largely for a huge proportion of all living organisms, this is actually maintained. Then we come to the translational machinery, whereby the tRNA is, first we talk about the structure of the tRNA. So like we've already mentioned, the tRNA is the major like executor of everything in the cell. So it has, its structure is majorly given two names. One, it is called as the clover leaf structure, and it's also called the L-shaped structure. So this is a point where you need to be careful when you're answering a question. It's also part of one of the assignment questions as well. So the clover leaf structure, because it looks like that leaf. If you just like look it up on Google, you'll see that that is how the leaf looks. It has three beautiful leaflets or petal-like structures. This is in two dimensions, but the three-dimensional structure looks like an L. Its length, its different properties, like it has an acceptor arm, an anticodon loop. The anticodon loop is where it binds to the codon. It has an acceptor arm where it binds to the amino acid, a 3' terminal CCA terminal, then the D loop, often containing D, Psi, and C. And these are all of the different properties of, or all of the conserved properties of any tRNA for any organism. Then the exceptions, there are some exceptions where you'll see multiple tRNAs for the same animal, like for glycine, for the same amino acid, like for example, glycine. The second major component of the translational machinery is, of course, the ribosome. And ribosomes are ribonucleoproteins. They have ribonucleic acids or rRNA, like we discussed in case of processing as well, and of course a large number of proteins. It's a macromolecular complex, and it has majorly, it has two subunits, 50 and 30S. For prokaryotes, mitochondria and chloroplast has the 70S ribosome, which has 50-30 subunits, and these are the huge number of proteins. In case of eukaryotes, you have an 80S ribosome. The S here is for the Swedberg unit, which we have also discussed previously, which depends on the sedimentation coefficient and is somehow related to its size as well. And in that case, when the ribosome is different, in that case, the constituting subunits also differ. They are 60S and 40S respectively. So the 80S ribosome is described here. The other contributors are in terms of the processes. So here we look at the process. If you look at the mechanism or the process of translation, the first and most, I would say, the most crucial and the most controlled step is the activation of an amino acid or the charging of a tRNA. You have a tRNA, you have to make sure, like the cell makes sure that the correct amino acid is added to the correct cognate tRNA. So that charging step will decide whether an organism will have a functional protein and will be able to execute one particular life process or not. It is that crucial. So this charging step is taken care of by an enzyme which makes next to no mistakes, which is the amino acyl tRNA synthetase, which is responsible for this charging step, and it is extremely crucial. Then here we have mentioned what are the first amino acids. In case of microbes, it is the N-formyl methionine, FMET. Usually it is written as FMET, and here it's different. In case of FMET, there is an initiator tRNA which is different, and in case of eukaryotes, it's a different scenario. Then the initiation step, it involves the Schindel-Carnot sequence and an initiation codon, and based on that, the ribosome will be, it actually recognizes the Schindel-Carnot sequence and it binds. Then the initiator tRNA with the FMET, here you have the initiator tRNA which carries the correct amino acid, not a normal methionine, but a formyl methionine, charged methionine tRNA, methionine tRNA, it binds and it executes the GTPase activity. Then the second charged tRNA comes in, it enters the A site. You have the EP and A sites in the ribosome. So the second charged tRNA enters the A site, and then extension can be carried out. Here we look at some of the differences. Majorly, it is with respect to the start codons, number of initiation factors, the sequences that are recognized, like for example, Kozak sequences for eukaryotes and Scheintel-Garneau sequence in prokaryotes. Then the process of initiation can differ. Then in the process of elongation, you have several factors like EFTU, TS, and EFG. Then there is decoding, formation of the peptide bond, and translocation, which basically means the movement of the ribosome along with the movement of the tRNA is like their movement with respect to each other. Let's put it that way. That is the translocation. You shift from the first three to the next three. Sorry. Um, then the factors involved in elongation were discussed. The process of peptidal transfer, they carried out by the enzyme peptidal transferase, which is why the ribosome is often also called as a ribozyme, because it has this enzymatic activity where it is able to synthesize a peptide bond. And the peptide bond involves these formation of this ester, or this conversion of this ester bond into the amide bond, and it extends the polypeptide chain. Then this process of translocation needs to be carried out, where the RNA which has been deacylated, it is removed from the P site. The peptidal tRNA moves from the A to the P site, and ribosome should move one codon down. So this is carried out by EFG factors, and finally comes the process of translation termination, which is where release factors RF1 and RF2 are involved. They bind to the ribosome near the A site, and the release factors are removed, and then the 70S ribosome, because it is unstable by itself because of some initiation factors like IF1, IF3, and RRFs, which are ribosome recycling factors. This 70S ribosome, it dissociates into the 50S and 30S subunits so that it can again undergo this is actually known as ribosomal recycling, which ensures that after a ribosome has done its job and it has synthesized one particular polypeptide, it should again be available for synthesis of either the same or a different polypeptide. So it must undergo recycling continuously. So that is what we see there. And under post-translational modifications, we have phosphorylation. Just a second. Yeah, we have phosphorylation. We have where a phosphate group is basically added. It is discussed in quite detail how this phosphate group is added, PO32 minus group is added. It is an extremely commonly done post-translational modification, but yes, it exists. Then comes the glycosylation step, which is a part of which is taken care of by the ER and the Golgi apparatus, and approximately half of all the proteins synthesized within a cell undergo this process. Then there is ubiquitination, which is where a ubiquitin moiety is added, and it marks that this particular protein, for whatever reason, it could be incorrectly synthesized, it could be carrying some sort of a defect, a mutation, or for whatever reasons, if it is if the cell is in a position where it needs to conserve energy, it needs to break down a protein and get amino acids, or to regenerate its amino acid pool, or any other reason. Under those circumstances, that protein can be marked for degradation, and that degradation is carried out via the proteasomal machinery, and that is where ubiquitination comes in. And that step was also discussed in detail, how ubiquitin is first activated, it interacts with ATP, forms the intermediate, then ubiquitin is transferred to the E1, then E1 to E2 it is transferred, and then there is E3 ubiquitin ligase which comes in, and then polyubiquitin is attached to the substrate. Then it is carried to the proteasomal machinery, which it's like a cylindrical structure with a cap at the top and the bottom, and this polyubiquitin marked protein is brought to that proteasome, goes inside. It has a lot of enzymes which digest it and digest the protein, remove modifications, digest the polypeptide or protein, and it releases each of the amino acids into the cell for their recycling as well. So one common thing that you would observe here is that any resource within the cell, it must undergo recycling. It's a crucial part of survival. And other post-translational modification finally includes methylation, which one of the major contributors for this is SAM, or S-adenosyl methionine, and the enzymes that actually do it are methyltransferases, and majorly you can find their role in epigenetic modification, in silencing, and yeah, epigenetic control is largely taken up by methylation as well. Like that is for DNA, of course. This we are talking about proteins, but then yeah, methylation as a modification itself is very, very crucial. So that kind of brings us to the end of the recap for this week. We will next look at questions, and we'll just take like a short break and come back to it. Okay, so we will now discuss some of the questions. So for this week's assignment, the topics that were covered, based on that, we'll just go through the questions. So the first question asked about the central dogma of life and what was the enzyme that was used for transcribing the genetic information. So obviously we discussed the process of transcription just now in quite detail. So here what you just because the options at one look they are very similar, so it's easy to make a mistake, but what you need to have clarity on is what is the template and what is being synthesized. So obviously the correct answer is DNA dependent RNA polymerase, because you are transcribing the genetic information. Genetic information is in the DNA, and based on that information you are synthesizing RNA. Second is, which of the following combination of events in the central dogma of life are possible to not consider in vitro experiments? So if you remember, in the next lecture notes as well as in the lecture videos, there was a table that showed what are the possibilities and like what are the normal conversions that occur, what are some exceptions, and what are other possibilities that can take place. So you can find a number of combinations here. So here you can see DNA to DNA, which is the replication process. This is DNA to RNA transcription, RNA to protein, which is the process of translation, and RNA to DNA, which is reverse transcription. Makes sense, there is no issue to that. But how are you eliminating the other possibilities? Here you are seeing that DNA to protein is being synthesized, which obviously here it's mentioned that you cannot consider in vitro experiments, and under normal circumstances within a cell, within a living organism, this the step of transcription cannot be bypassed, and DNA to protein under normal circumstances will not be synthesized. Whereas in the third case, sorry, there's a lot of background noise. In the third option, you see that there is protein to DNA, which is also obviously not possible. And the fourth one has a difference in protein to RNA. Yeah, this reverse process also does not take place. So there's one option which is wrong in each of them, just needs a little bit of careful exploration. Then in the third question, we have been asked that the Taq DNA polymerase, which was found from Thomas aquaticus in hot springs in Yellowstone National Park, works at an optimal temperature of dash with dash as the cofactor. So it was found at 167 degree Fahrenheit, if you remember. It it is functional, or it is, I wouldn't say functional, it performs optimally at the temperature of 72 degrees centigrade, and the cofactor it uses under these circumstances is Mg2 plus. MgCl2 is therefore used for setting up the reaction. Therefore, magnesium is the cofactor which is utilized under these circumstances. Then third is what is the antisense strand for the mRNA sequence, which is given here. So before you approach, okay, yeah, here the other options would be eliminated here. First elimination would occur based on the temperature where it is optimally functioning. You need 72 degrees for the PCR reaction. Where polymerase actually functions, you have higher temperatures of 92, 96 degrees for the denaturation step, but the elongation or the extension or polymerization step, it occurs somewhere around 72 degrees. So that is the temperature, what should I say, the temperature efficacy you are looking at. So that is taken care of by these enzymes, and you can eliminate it also by the cofactors that are being mentioned here, because magnesium is the cofactor under those cases. In question 4, we were asked what is the antisense strand for this given mRNA sequence. So before you start off with a question like this, what first needs to be kept in mind is what is antisense, because these terminologies can often get confusing. Now, the way we had looked at during the discussion was that the coding strand carries the actual code, and it is the one that makes sense. So that is how we took up that approach, right? So that is the sense strand. The anti-sense strand is the template, which is taken up, which is taken as the reference, and against it you have the base insertion taking place. So for this case, so what do you understand from it? You understand that for an mRNA sequence which is given like this is the coding sequence, or this is the sense sequence. What you need is the antisense one, so it would be complementary to it. That is how you first start off with the approach. The second thing you need to look at is the orientation. That is also very, very important. So here, based on the orientation, what we will see is if this is 5' to 3', we would need a strand which is under normal circumstances 3 prime to 5 prime and would be complementary to it. So that would be, for you it would be A, U, or for a DNA it would be T, G, C. So you see ATGC here, you see ATGC here as well. If you look carefully, you will see that all of these other sequences are also the same, but the orientation is different. So this is where you need to have clarity that if this is the 5 prime to 3 prime mRNA sequence, if for example, consider this end to be the 3 prime end, if this is the mRNA sequence and this is the strand you are using as the template against which complementarity is being set, what do you simply see? This end is the 3 prime end, right? So starting from the 3 prime end, you would have ATGCC and so on. So that is where you get A as the correct option. So just in case, if just me explaining this vocally is somewhere at all complicated, what you can do, always draw it out, make figures and just make two lines and approach your questions, because that will avoid any confusion under any circumstances. Kind of continuing with this theme only, what is the mRNA sequence in question 5? This is what we were asked, that what is the mRNA sequence if the DNA sequence is this particular stretch of DNA in the sense or the coding strand. So the sequence of the mRNA would be the exact same as that of the coding or the sense strand. That is exactly what we will get. What you need to keep in mind is the orientation. So the sense strand ka orientation also is exactly maintained, because like we said, if this is the mRNA which is being synthesized, this is the anti-sense strand. So the sense strand would also be complementary to it. So they would be exactly the same. That is why I'm saying the sense is maintained, the code is maintained, which is where the name is coming from. So you would have the 5 prime to 3 prime orientation with the exact same sequence. So that is what we see here in option C, and that is how you come to the conclusion. Sorry. Yeah. In question 6, we were asked that if you have an assertion that in intrinsic mechanism of transcription termination, the rho factor is not essential. The presence of GC-rich hairpin followed by 7 uridine residues is required. So if you just look at statement A or the assertion, it is correct. Assertion simply means a statement, a positive statement, right? So in this case, that is correct. You don't need rho factor, and the basis of the intrinsic termination, transcription termination mechanism is the formation of this GC-rich hairpin followed by a stretch of U residues, poly-U. Poly-U stretch is how it is usually called. So that is absolutely correct. In the second statement, we are being given a reason. So this formation of hairpin structures which have weak RNA-DNA hybrid interactions signals the dissociation of both the polymerase and the transcript. So in statement 2, which is the reason, we are saying that hairpin structures ka formation, it is occurring. They have weak RNA and DNA hybrid interaction. So because the basis or the what should I say, the logic by which this is actually occurring, that intrinsic termination is occurring, is simply because the strength of the RNA DNA hybrid interactions is less compared to that of the GC hairpin, which is why the dissociation is taking place. So the statement R, or the possible reasoning which is being given, is actually explaining the statement given in the assertion, and therefore among the given options, option B would be correct, because it is saying that assertion and reason both are true, and in fact the reason is the correct explanation of the assertion as well. So that is what it is confirming. Then in question 7, we were asked that rho factor is an ATP dependent helicase which is required for the termination of transcription. Rho protein is a dash. So this is like absolutely a direct question. A dimer, trimer, tetramer, or a hexamer are the options that have been given. And rho, as we have previously discussed also, is a hexameric protein. It kind of rolls along the nascently synthesized RNA, and it chases the polymerase, and it basically pulls, like if you think of it as a pulley kind of a system, it pulls the nascent RNA towards itself and away from the DNA, allowing its release to actually take place. Then comes the eighth question, where we were asked which transcription factor recognizes the Tata box in eukaryotes. So here we talked about the sequence in which these each of these transcription factors, they came in, they assembled, and they formed that entire transcriptional machinery, right? So among those, the first and probably one of the most important steps, kind of considered to be the priming step for transcription to proceed in eukaryotes, that is executed by TF2D, which is the first transcriptional factor that comes in the machinery. So TF2D is the answer to that. Then in question 9, yeah, we were asked that in the post-transcriptional modification of mRNA, when is capping occurring, right? So if you remember in our just very recent recap as well, that capping it, so you do not need the entire mRNA to be synthesized for capping to be actually carried out. Yeah, you do not need the entire mRNA for capping to be carried out. Instead, what you would need under those circumstances is just a nascent mRNA to emerge, and that itself would kind of direct its future localization as well. So the process of capping, it initiates as soon as around 20 to 30 nucleotides, which is what the question or the first option also shows, that as soon as you have 20 to 30 nucleotides that are formed, capping process, the MGPP addition, moiety addition, it takes place. And after that, once the entire mRNA has been synthesized, after that you would have the attachment of the cleavage and polyadenylation steps, because that is a part of tailing, and you obviously need the entire mRNA to be synthesized under those circumstances. So that is when that part would occur. Then in question number 10, we have been asked that poly A polymerase, the enzyme which adds the poly A tail at the 3' end of the mRNA, is a template independent DNA polymerase, template independent RNA polymerase, RNA dependent RNA polymerase, or RNA dependent DNA polymerase. So first of all, if you remember, PAP or poly A polymerase is a template independent enzyme because it just simply adds adenine residues, right? It adds A residues one after the top of the one after the other. Now the point of confusion that could come in, it's not very confusing, but something that could come in is that adenine is common to both DNA and RNA. Like if it was uridine, there would be no confusion at all. But for adenine, what happens is that you need to keep in mind what is being synthesized. You are tailing an mRNA. So in these circumstances, it is a ribonucleotide which is getting added, right? An RNTP, not a DNTP. Therefore, the enzyme which is working, it is template independent, but it is an RNA polymerase, not a DNA polymerase. So these are like very minute like errors that we often make without even realizing, like in the hurry of trying to finish a paper or something like that. So what you need to do is you need to be mindful about things like this. um, then coming to question number 11, which of the following amino acids are coded by one codon each, only by one codon each? So we looked at the entire genetic code, right? And while most of the properties are conserved for all of them, there was specifically this part was highlighted that there are only two amino acids that are coded by a single codon, and those are tryptophan and methionine. These two only have one codon coding for them. So this is like an absolute direct question, no ifs and buts about it. Leucine here is coded by like I think 6 codons. Lots of degeneracy is seen for leucine particularly. Then in question 12, we were asked which of the following is not a property of the genetic code. A, some of the start codons are GUG and UUG. There is no comma in the genetic code. Third, the genetic code is degenerate. And fourth, it is overlapping. So start codons in some cases can be GUG and UUG. So it is a property of the genetic code. There is no comma in the genetic code is correct. Yes, it is contiguous, which means it's one after the other. It is degenerate, absolutely correct. And fourth, the code is overlapping is not correct. It's a non-overlapping code, which is what we said, that after the first three nucleotides are corresponding to an amino acid, the next three come in. So there is no overlap between these nucleotides at all. Then in question number 13, we were asked that tRNA or transfer RNA is usually of dash ribonucleotides in length. So if you remember the different properties of tRNAs that were discussed, you would know the answer to this, and it is about 75 to 96 nucleotides, like around 70 to 90 nucleotides is the average length. So that is the answer to this question. Then in question 14, yeah, this actually brings up one point we have already discussed about how you can often get confused in very similar options. So when you just talk about the tRNA and its shape, it has both these terminologies. It is both clover leaf and it is L-shaped. But always keep in mind that clover leaf is the two-dimensional structure, which is just where if you have the sequence of the tRNA, that is what its 2D structure would look like. But when it folds and when it takes up its 3D structure, it has that L sort of shape that gives it its final three-dimensional structure. So make sure you do not make and also if you just look at the options, it can be considered to be V-shaped as well, because this is pretty much what its shape looks like. You can consider it V, but then it is said to be an L-shaped structure, and that is something you should have clarity about. Then finally, in question 15 for this week's assignment, we were asked that which of the following set of combinations is correct regarding the tRNA molecule. So this is again a property, like it was mentioned under the properties of tRNA, transfer RNAs. So I'll just directly get to the correct answer, which is option B, where you have a 3' CCA tail. We talked about it, that the 3' end needs to be, the CCA needs to be added to the 3' yeah. So a 3' CCA tail needs to be added to a tRNA as part of the modifications. Then the T-arm, it has the Psi. Psi is a modified version. So Psi or pseudouridine, it is a modified amino acid. Or rather you can say a modified nucleotide, not an amino acid, it's a nucleotide. It's not like the uridine residue that we normally know of. It has some changes in that, in its regular structure, and that gives rise to the pseudouridine. And thirdly, we have the D loop, which often it contains the dihydrouridine structure as well. So this covers the assignment for this week that was given. So I hope most answers were correctly approached. And as part of the upcoming topics that would be taken up, which were for week 7, which we will discuss in much more detail in the live interaction session on Monday. As part on 20th March, we'll discuss it in more details, but just to kind of introduce the topic, and not even introduce the topic, just goes through some questions related to the same. So as part of week 6, we have looked at some of these cellular processes, like part 1, where we looked at the central dogma, PCR, transcription and translation in quite a lot of depth, and replication, of course, also. That was the focus for week 1. Week 2 is, it kind of takes a different approach. There is a lot of focus on the immunity, like the immune system and how that operates, what are the different players in it. And also towards the later lectures, we also look at some of the other processing steps that go in, like for example the modification of proteoglycans, of other vesicular trafficking, what happens after a protein is after an mRNA is transcribed, where does it go, what happens to a protein, how is it directed to the correct compartment, and so on and so forth. So in reference to that, we have a few sample problems that are kind of related to these topics. So first question is about B cells. So B cells are involved in which of the following types of immunity? So as we know that the entire immune system, or immunity in general within eukaryotes, within humans, let's say humans for example, for now, it can be majorly divided into CMI and HI. CMI is cell-mediated immunity. HI is humoral immunity, right? So humoral, it comes from the word humor, which is related to the antibodies. So when antibodies are executing some part of the immune, when they are regulating the immune response to a particular, what should I say, so to a particular invader or pathogen or some sort of infection. When an organism is suffering from some sort of an infection, when the antibodies are the response to it, that is known as the humoral response, right? Whereas the cell mediated response usually involves, as the name indicates, cell mediated immunity. It involves a lot of cells, other cells, which have a role to play in it, which involves majorly the T cells. Then why are B cells not included under the same? That is because B cells are responsible for releasing their membrane receptors, and their membrane receptors are basically the antibodies, right? So that is why B cells are involved in the humoral immunity. If you look at the other options, they are obviously not corresponding to it. Cell-mediated immunity is not the answer to it. APC are antigen presenting cells, which are different, and acquired immunity is something completely different. Acquired, like we talked about the types of immunity. There you can also classify immunity in a different angle, where um, the immune defenses that an individual is born with, those are innate. Very broadly speaking, that those are innate, included under innate immunity, whereas the kind of immune defenses that come up over time due to response that has been generated within that same individual. Like for example, if in childhood you suffered from one particular disease, some sort of infection you suffered from, and your body responded to it, then your body remembers that, okay, that is what an infection had come in, and this was the response that I elicited against that. When that memory is kept within an individual, within their immune system, within that individual, then that is acquired immunity, which you are acquiring because of an insult that the individual's immune system um, had to face at that age. So that is why that approach is completely different. We will discuss this in more detail in the next class in 3 days. Then the next question looks at the bond that is involved in joining the heavy and the light chain at the hinge region in antibodies. So if you look at the structure of an antibody, you see that it's Y-shaped, right? You have two heavy chains and you have two light chains. Now, how are these four different polypeptides, or four different not proteins, they are polypeptides actually. How are these four chains at all connected to each other? So the covalent linkage that actually connects these chains with each other is the disulfide linkage. So even between the heavy chain and light chain, there are disulfide linkages, and between the two heavy and light chains also we see disulfide linkages. So the answer to this is the disulfide bond, which is why if you use a reducing agent, you will get four different polypeptides. Or while the heavy chains are of similar molecular weight, effectively there are four polypeptide chains. Oops, sorry. The third question is about the antigen binding site on an antibody. So what are the options here? We have epitope, paratope, antitope, and isotope. So let us first remember very with like absolute clarity the difference between an epitope and a paratope. The epitope is the protein's some part of the protein, any part. It can either be on the surface, mostly it is on the surface. It can either be the continuous stretch of nucleotides, like nucleotide, say n plus 1, n plus 2, n plus 3, n plus 4, one after the other. These nucleotides all together, they are not nucleotides, sorry, amino acids. These amino acids of a protein or of an antigen in particular, they are forming a region, say some sort of a cleft. They are forming a bump on the surface of the protein, and an antibody is binding to that bump exactly. Then it is binding to that epitope. Now on the if you consider this to be the protein and this is the bump, this is the epitope, antigenic epitope. This epitope is recognized by an antibody. Imagine this to be the antibody. It's Y-shaped, right? So when it is binding on part of the antibody, the regions which bind, which would be these two entities which are formed by heavy chain and light chain, that is the paratope. So the antigen binding site on the antigen, on the antibody is the paratope, whereas the site on the antigen where the antibody will bind is known as the epitope. I hope I didn't confuse you. So these are the differences between the two. Then in question 4, we have been asked about the MHC class, which is involved in antigen presentation in macrophages. So MHC are a major histocompatibility antigens. These are a class of antigens which have a large number of roles, but yeah, they are sort of membrane receptors which are majorly involved in histocompatibility and in the immune system as well. They are also responsible for presenting antigens on the surface of these cells so that they can be recognized by other cells that will elicit an immune response against it. So the two classes or two types of MHCs are class 1 and class 2. Among them, class 2 is actually involved in antigen presentation for the APCs, antigen presenting cells, which includes macrophages, B cells, and dendritic cells. But here, since we have been asked about macrophages only, the answer to that would quite directly be MHC class 2. Then in question 5, we have been asked that the reagent which is used to screen the clones in hybridoma technology for the production of monoclonal antibodies is which of the following. So if you've gone through those lectures, you would know that for monoclonal antibody production, you need this hybridoma technology, and that is done by a HAT mechanism or HAT assay. So what is done there is the reagent that is used. That is what we are being asked for here, right? For HAT. So if you just look at the option, you will see two very similar options, but then yes, again, these are just given to confuse you. But then the HAT medium is actually hypoxanthine, aminopterin, and thymidine. These are the components that are present in it. Aminopurine is not the answer to this. So very slight difference between the two, but yeah, that's something you will have to be a little careful about. Then in question 6, we are being asked what would be the blood group if no agglutination is observed with anti-A and anti-B antibodies. So if you look at the like the way in which these blood groups are classified, A, B, O, and AB, blood group A, B, O, and AB, these four, then one of the ways in which it is understood is where and under which circumstances they would undergo agglutination. So agglutination basically means sort of clumping because there is a antibody which is binding to the antigen. So for A, we have anti-A antibodies would bind to or would cause agglutination. For B, anti-B would cause. For blood group AB, both would cause, but when there is blood group O, these individuals do not express A antigen or B antigen on their RBCs, and as a result of that, either anti-A or anti-B antibodies do not cause any agglutination. It's a very simple question. You just need to keep that in mind, and the mechanism behind this is also something that needs to be studied, which is like why at all are O antigen people with blood group O are not expressing this A and B. So there's an enzymatic process which is different in these individuals, making them donors. Actually, this is why they're used as blood donors. They won't cause agglutination or a adverse immune response in other people under most circumstances. Yes. Then in question 7, we are being asked that the agent which causes the fusion of the spleen and myeloma cells, which is what is actually done for the hybridoma technology, which we discussed here about HAT medium. So for that, what do you use to actually form the hybrid, to fuse the spleen and myeloma cells? So the answer to that is the use of PEG, which is polyethylene glycol, PEG 4000, PEG 5000. These are different versions of, not versions, different molecular weights of these different chemical reagents. So most commonly, PEG is used for this purpose. So that answers question 7. Then in question 8, we are being asked that the biochemical cell surface marker for detecting apoptotic cells in flow cytometry through an exin 5 is A, phosphatidylserine, B. Phosphatidylcholine, C. Phosphatidylinositol, or D. Ceramide. So if you remember, during the lecture where we were, not lecture, during the interaction session, where we had discussed about the different types of biomolecules, and you talked about nucleic acids, we talked about lipids and many more. So there, we had gone through this part as well, where we had said that when a cell is undergoing apoptosis, apart from its many different changes that it is undergoing inside the cell, one major signal that it brings to the surface is that if you consider the plasma membrane to be, not you consider it, because the plasma membrane is a bilayer, a lipid bilayer, you have two layers like this, right? So if this is the outer leaflet which is facing the cell exterior, and this is the inner leaflet which is facing the cytoplasm of the cell, then under normal circumstances within a healthy, properly functioning cell, what you will see is that the phosphatidylserine, which is obviously a phospholipid, is localized exclusively to this inner leaflet towards the side that faces the cytosol. But when a cell is undergoing, like when a cell has decided that it is not going to invest more energy and more resources into surviving, because for whatever reasons it has suffered a lot of stress, it is carrying mutations, it is not helpful for that cell to survive, and it should not survive. When that is the situation, sorry, when that is the situation, then what we see is that there is a difference, there is a shift, or there is a localization of the phosphatidylserine from the inner leaflet to the outer leaflet takes place. Just give me a second. From the inner to the outer leaflet, phosphatidylserine is shifted, and as a result of that, you can detect such cells using an exin 5 staining in flow cytometry or flow cytom FACS or flow cytometry associated cell sorting process. Then after that, we look at question number 9, where we have been asked that which of the following applications is not possible with flow cytometry. So A, isolation of green fluorescent protein from plants. B, in identifying various cell types unique to certain diseases. C, in immunophenotyping. Or D, in determining amino acid sequences in a protein. So you can't do anything, you can't detect amino acid sequences using flow cytometry, right? It is majorly used for cell surface markers. It is used for antibodies, for identifying cell types unique to certain diseases. So this is not a possible application of this technique, and therefore that would not be the answer to this question. Then in question 10, the channel that is attributed to the granularity of the cell in flow cytometry is A, forward scatter, B, side scatter, C, upward, or D, downward scatter. The answer to this is side scatter quite directly, majorly because the forward scatter is, it kind of depends upon the cell's size, whereas the granularity of the cell, it brings about the size, it is brought about by the side scatter. Then in question 11, the dye that is used to analyze cell cycle using flow cytometry is A, propidium iodide, B, propidium bromide, C, crystal violet, or D, phycoerythrin. The answer to this is PI. So PI or propidium iodide is most commonly used for flow cytometry. In 12, we are being asked that the cellular organelle which is involved in liver detoxification is A, smooth endoplasmic reticulum, B, rough endoplasmic reticulum, C, lysosomes, or D, the nucleus. The answer to this is the SER, which is the smooth endoplasmic reticulum that is used. Then the class of pumps which are used to maintain acidic pH in lysosomes is A, the calcium pump, B, the F-class pump, C, V-class pumps, or D, sodium-potassium pumps. So the answer to this, for pH maintenance, for acidic pH maintenance, V-class pumps, they come in and perform this function. In question 14, we've been asked that the organelle that adds proteins to glycosaminoglycans to form background noise to form proteoglycans is A. Lysosomes, B. Golgi complex, C. Smooth endoplasmic reticulum, or D. Rough endoplasmic reticulum. The answer to this is B, which is the Golgi complex. Then in question 15, we've been asked that the sorting of the newly synthesized proteins to their respective cellular destinations is called A. Protein export, B. Protein import, C. Protein targeting, or D. Protein delivery. Then the answer to this would be protein targeting. Basically, it's the process where after protein has been synthesized, you are making sure that it is sent to its correct destination. And for that, you are targeting the protein to its right location, right? So that is why protein targeting is the answer to this. Question 16, we've been asked where does the ribosome and polypeptide chain complex get targeted once a short segment of the polypeptide chain is synthesized during protein translocation. So after you have a short segment which has been synthesized, you will send it to the ER, like the cell sends it to the endoplasmic reticulum. The binding of the signal recognition particle, which is SRP, to a larger subunit during translocation leads to which of the following? A. Translation arrest, B. Transcription arrest, C. Progression of transcription, or D. Progression of translation. The answer to this is that it leads to translational arrest. So because the large subunit is of the ribosome, so when SRP or signal recognition particle, it binds to the larger subunit, translation at that point is arrested or is abruptly it stops. It is stopped from progressing at that point of time. Once it has been localized to the translocon present within the ER, again it would be resumed under those new conditions. In 18, we are seeing protein glycosylation. What is that process? It is the addition of protein to proteins, addition of lipids to proteins, addition of carbs to proteins, or addition of lipids to carbohydrates. So the answer to this is addition of carbs to proteins. Protein glycosylation means you have a protein, you are adding a carbohydrate molecule to that protein and carrying out its glycosylation. In 19, we've been asked that the class of lipids that transport the dolicol and sugar from cytosolic site to the ER lumen site is, you have, like we talked about phosphatidylserine as well, where it is being removed from the inner site or the inner leaflet to the outer one. Here also you are flipping it from the inner to the outer side. So the enzyme which actually does this, not enzyme, the lipids which actually do this are called as flippase. Yeah. And then final question 20, we are looking at the transport of proteins from the endoplasmic reticulum to the Golgi complex is mediated by A. COP1 vesicles, COP2 vesicles, synaptic vesicles, or the lysosome. So from ER to Golgi is it is the anterograde transport, which is in the normal direction that you expect it to proceed, whereas from Golgi to ER would be the retrograde transport. So anterograde transport is carried out by COP2 vesicles, and therefore that is the answer to this question. So this is where we are going to close for today's session. So what we have discussed today, we have first gone through all of the topics that were discussed in week 6, which were quite extensive, mainly centered around the cellular processes, which were starting with replication, first starting with the central dogma, replication in particular. Then we moved towards PCR. We looked at transcription in quite depth, and we finally looked at translation. After that, we have discussed some of the assignments for this week and for week 6, and I'm pretty sure you've already submitted the assignment for week 7, and we'll be discussing that in the upcoming week session as well. And with that would bring us to the second part of the basics of cellular processes, where we'll be looking at immunity and these protein transport, what are the different factors that are involved in this, and and we'll look at it in much more detail. So that pretty much covers our session for the day. I hope these are helpful for you, and you can come up with your doubts and queries, and if necessary, post them in the discussion forum, or you can join any session and bring up any question from previous weeks as well, and we'll try to address them and solve them as quick as possible. If not possible at that moment, we'll respond in the discussion forum, and we hope that everything, these interactions and all of the material that is being provided to you is helpful. And with that, I will conclude my session for the day. So thank you to those who joined, and see you again on Monday. So thank you.